Hello everyone, how you doing? This is Mr. Los. Welcome to another exciting edition of HO Chemistry Flipped Classroom. I hope this message finds you as well as possible. I'm recording this in March of 2020. School is currently closed for the coronavirus epidemic. Maybe you're watching this at some later point, but right now it is a crazy time, and I hope you folks seeing this are doing as well as you can be doing. So we're going to talk today about a couple of different types of pathogens, bacteria and virus, obviously relevant to the current situation. We're going to start off talking about bacteria and about how they work, and, as well as penicillin. And so this does focus on penicillin, so one of our questions is going to be, to what degree does, by what mechanism does penicillin help stop bacterial infections, and what are some problems associated with the overuse of antibiotics? You should have a note packet that goes along with this. So you don't have to write all these notes down. So people sleep on bacteria. Let me tell you something. Bacteria are incredibly successful creatures. Arguably, maybe some of the most successful creatures of all time. Certainly, bacteria or bacteria-like things have been around since life has been around. Of course, if you're talking about define success, right? We human beings like to think that we're the most successful, but we're setting our own rules of success, our own success criteria. If you're talking about actual number of individuals, of course, there's many more bacteria than every form of Earth combined. But even if you're talking about biomass, like what they would weigh, if you could collect up all the bacteria and weigh them, bacteria weigh about 35 gigatons. Whereas all other animals combined only weigh about 2 gigatons. So however you slice it, there's more bacteria than anything else. They've been around for a long time, and bacteria or things like them are going to be around long after human beings are gone. They're single-celled creatures. You can't see them. They're everywhere. There is more than likely more bacteria cells on you and in you than your own cells. You know, somewhere around 39 trillion. Now that number can vary wildly. The vast majority of them are either kind of neutral or helpful, actually. There's a bunch of bacteria in your stomach and in your whole digestive tract that help us to digest various foods. Some people say many health things are affected by these bacteria. Who knows, but it's interesting to think about. And they belong to a class that we talked about, I think, a little bit when we did our biochem review. They're called prokaryotes. They have no organelles, and they have cell walls, which turns out to be pretty important. Just a couple of things. Here we have plant cell, animal cell, bacterial cell. The main thing to take note of here is that they have cell walls, and they don't have these membrane-bound organelles that we think of in eukaryotic cells. So penicillins are antibiotics produced by a type of fungus, and they were discovered by chance by this guy Alexander Fleming in 1928. He was growing bacteria on a petri dish, something like this up here. Obviously, this is kind of a recreation, and he noticed that some mold had grown on there, sort of contaminated his experiment, and then he also noticed that right around where the mold was growing, no bacteria were growing. And the big kind of insight was, instead of just like throwing it away or getting mad that his experiment was ruined, he kind of took note of that and ended up realizing it was this mold called Penicillin notatum. He didn't identify the exact substance that was causing this inhibition, but he is given credit for discovering penicillin. And then the actual drug penicillin, these guys Florey and Chain figured out. They isolated penicillin as the actual component that was inhibiting the growth of the bacteria, and they're the ones who really paved the way for it to become an actual medication. And we take this for granted. It's a huge, certainly one of the biggest moments in all of human history was the development of penicillin. Before penicillin became widespread, you would have a simple maybe infection and die from it. Get a cut on your foot, get a bad infection, and next thing you know, it becomes a very, very serious thing. So this was absolutely huge. It can't be overstated how big of a moment this was in human history. One of the biggest. And so here we have the structure of penicillin. Thiazolidine is this portion of it right here, and then the really big important part is called the beta-lactam ring, which is this 4-membered ring here, 4 carbons. We got a carbonyl right here, and then the nitrogen. So 3 carbons and a nitrogen. So it has tight bond angles. Now, if you think about it, this is a square, so those angles would be 90 degrees. That is away from the typical bond angles that we would think of in singly bonded carbon, which is sp3 hybridized, and also in doubly bonded carbon. So those bond angles are much tighter than a carbon would like, so to speak. So that means the beta-lactam ring is unstable, and it's prone to becoming unsprung. So there are a few varieties of penicillin, and this R group right here varies depending on the exact type of penicillin. Oh, I should say, by the way, it's clearly not in red here, but this is the beta-lactam ring right here. So basically, this beta-lactam ring, the tight bond angles, it comes loose. Basically, the ring breaks, and that is what's responsible for the action of penicillin. So bacteria have cell walls, and they are constructed, of course, by the bacteria as it's reproducing itself. Transpeptidase is an enzyme that bacteria uses to help create cross-linkages in its cell wall. And basically what penicillin does, oh here it's red in this one, what penicillin does is interfere with that enzyme functioning, and basically the bacteria can't make their cell walls effectively, and they're going to die. And this, before we go on to this highlights, or something that's important about drugs is, if you're going to try to kill some sort of pathogen that's inside of you, you have to have the ability to attack something that thing has, but not you. Now, you can drink bleach. That will definitely kill any and all bacteria that the bleach comes into contact with. The problem is, of course, bleach is incredibly, is quite toxic, or we've talked about how toxic is kind of a word that's tough to define, but it's bad for you. You shouldn't drink bleach. So we have to be able to find something that can attack the pathogen but leave the other cells unharmed. It's a tricky business, but in the case of bacteria, because they have a cell wall and our cells don't, that's one thing we can target in terms of dealing with bacteria. So one thing you should do is just check in here for a second and label these 3 areas just so you make sure you can identify those before we move this on. So, as I said, this was a momentous moment in history when penicillin was developed, and then eventually other antibiotics that are different than penicillin. But what started to happen is bacteria started to become resistant. Penicillin, and there's various penicillins, but various types of penicillins and other antibiotics started to not work. And basically, it goes like this. Bacteria are very quickly reproducing creatures. You could have many generations of bacteria in a week, so they evolve very quickly. But let's just say that we have some normal bacteria here, and one member of this group is kind of resistant to bacteria, or rather to penicillin or some type of antibiotic, just through a mutation, a random mutation. Well, you take the drug and you do kill off all of the ones that are most susceptible to the penicillin or to the antibiotic, but sort of by definition, the most susceptible ones are going to die, and the ones that are most sort of able to fend off will grow. And so what can happen is you kill off all the bacteria that are very susceptible to the antibiotic, and then you leave yourself with a population that are resistant to it. And over time, this can become a problem. And there's definitely certain types of bacteria where more and more of our antibiotics don't work on it anymore. Now, every time you use an antibiotic, the possibility of resistant strains developing is increased. And they're not going to, it doesn't happen to where, basically, if you never use antibiotics, then bacteria can't develop resistance to them, right? So that's why we want to restrict it to the most necessary cases. Penicillinase is an enzyme that can break down penicillin. So some bacteria can evolve the ability to break down penicillin. Bacteria also have an ability called, they can swap these things called plasmids. Plasmids are little circular loops of DNA. And certain bacteria, they can approach each other, they can exchange plasmids, exchange little circular loops of DNA, and maybe in that little DNA is, oh, here's the instructions to make penicillinase. And of course, they don't, necessarily, they're not thinking about this, but this is just something that is happening. And if there is a population of bacteria that have the ability to swap plasmids, one of those plasmids being antibiotic resistance, that population is going to tend to grow or be more prevalent in the face of antibiotics. So the plasmids and just sort of natural random evolution can increase this antibiotic resistance among bacteria. So what are some things that human beings are doing that are contributing to this? Well, one thing is overprescription of antibiotics. Now this, I think in recent years has definitely gotten a lot better, but you come into a doctor's office, you have the cold, antibiotics can't do anything for a cold because colds are a viral illness. And especially back in the old days, the doctor would just give you some antibiotics, maybe just in case, or maybe they just want to get you out of their office, like, hey, stop complaining, here, I'll give them these pills, at least they'll leave and I can go see the next person who I can actually help. So people giving antibiotics for no reason basically anything we're doing that is unnecessarily giving antibiotics is bad, right? So if you're giving somebody antibiotics who has the cold, that's a waste and contributes to antibiotic resistance. There is, and now with this COVID paranoia and all that, there are antibiotic soaps and all that stuff. Especially antibiotics and soaps, totally not necessary. Regular soap is just fine. You really don't need antibiotic agents in soaps. In fact, most of the time, unless you put the soap on your hand and let it sit for 5 minutes or whatever without washing it off, it's absolutely useless. Doesn't make any difference. But yet that contributes to antibiotic resistance. They give antibiotics to animals, as a prophylactic or as just a preventative measure. They're like, let's put some antibiotics in the feed, and that way, none of them will get sick in the first place. Again, all these things that we're just giving too many antibiotics, and presence of antibiotics all contributes to antibiotic resistance. And then patient non-compliance. So if you have ever been prescribed an antibiotic, and again, I think doctors these days are actually very resistant to prescribing antibiotics unless they're really necessary. But if you have been, they will tell you, take this for 10 days or 5 days or whatever, and take all of it, even if you feel better, or whatever is wrong with you clears up. The reason for that is because, let's say you take it for 3 days and you stop because whatever is wrong with you is improved. Well, again, by definition, what you've done is you've killed off all of the bacteria except the ones that are most resistant to that antibiotic that you're taking. You've left alive the most resistant portion of the population. What you should do is keep taking it, and the bacteria may not be completely resistant to the antibiotic, but so if you take the full course, you'll eventually kill all of them, or basically almost all of them. But if you stop early, you're leaving alive the most resistant population, so that is a problem. There's also penicillin in the water supply. There's actually a problem with all kinds of drugs in the water supply because, as we've kind of talked about with bioavailability and stuff like that, a pretty high percentage of drugs ends up passing right through you. It might end up being excreted through urine and all that sort of stuff, which is going to end up, it ends up out in nature. The matter cannot be created or destroyed. So if there's a penicillin molecule floating around in the water, it's out there, right? And again, anything that has more penicillin out there contributes to resistance. Many people you shouldn't throw any medications away, but throwing medications down the toilet or throwing them in the landfill is probably not a good idea because it puts antibiotics where we don't want them. Some other problems with antibiotics is, like we were saying, a good chunk, most bacteria are either helpful or neutral. When you take antibiotics, those helpful bacteria are going to die as well. So that can contribute to some digestive problems. But even if they're sort of neutral bacteria, if you have, we have this huge population of bacteria, most of them are just sort of living on us and doing their thing, but what they are doing is they're taking up space and they're competing for space and resources. If you kill those neutral bacteria, what that does is provide an opportunity for a harmful bacteria to come in there and occupy that space. So the neutral ones can just sort of crowd out the harmful ones just by being there. You can get allergies to antibiotics, including penicillin and some other ones. Here's one of the things you can do to help with antibiotic resistance. You can make a new side chain. You can modify that, and then that will sometimes a bacteria that can deal with one side chain that our group cannot deal with another one. It also might help allergic reactions. Before we go on to viruses, as we talked about a little bit though, Well, this is becoming a serious problem, antibiotic resistance. I talked about how drug companies like to develop medicines for chronic problems that people have their whole life. They don't really like to develop drugs for temporary problems like a bacteria infection. So there's also a money motivation that is sort of stopping, the development of completely new antibiotics and stuff because they don't want to invest the money. They'd rather develop like a new depression drug or whatever, some other chronic illness where someone has to take their pill the whole life. And again, blame capitalism, but it makes sense from an economic point of view that a drug company would not want to develop an antibiotic. Okay, so antivirals. Here's the part that we're all hyper attentive to right now is viruses and the coronavirus outbreak that's currently happening. And viral infections are much more difficult to deal with than bacterial infections, as we will get to in a second as to why that is the case. Bacteria are really small, even compared to viruses. I mean, viruses are really small, so are bacteria, but viruses are even smaller. You can't even see viruses with a microscope. They're sub-microscopic. There's viruses called bacteriophages that specifically attack bacteria. Viruses can't reproduce on their own. Bacteria can. Viruses can't. They have to hijack another cell's machinery. Viruses are assembled inside host cells. There's not much to them. There's basically a protein coat and some kind of genetic material. Sometimes there's a lipid membrane to them, but very, very little to them. They're generally not considered alive. Because of the fact that they can't produce on their own, they don't have any sort of metabolism. So for those two reasons, they're really not considered alive. They're pretty amazing things. You've got this molecule DNA. It happens to have the property that it can make a copy of itself, and sort of by definition, the strands of DNA that are best able to copy themselves are the ones that go on, and then the DNA ends up making more and more complex things around itself till you get something like a human being. But still, at the end of the day, what the DNA wants is to make more of itself. Or you could go the other direction. You could be something very, very simple relatively to a human being, like a bacteria. Or you can even be something even simpler, a virus, which is basically just a little strand of DNA with the bare minimum of stuff around it, where that DNA can manage to copy itself. So this is just that you see a bacteriophage there, and then you see the HIV virus. Viral illnesses, smallpox, polio, measles, all things that are mostly eradicated, except I'm not going to get into this right now. There's people who don't believe in vaccination. So measles, for example, is making a bit of a comeback. Colds and flu are viral. These days, I don't think people are underestimating the flu, but sometimes people do. It kills about 500,000 annually worldwide or more. Spanish flu killed about 20 million in the 1918 to 1920. Warts and herpes are viral things. AIDS, Ebola, and then, of course, the coronavirus, which causes the disease COVID. Most viruses use DNA for genetic material, but some use RNA, most notably HIV and coronavirus. Of course, I just added because I made this long before all this stuff happened. So RNA use an RNA blueprint to make a DNA strand, and we talked a little bit in our review of biochem. This is the opposite of what normally happens. They're called retroviruses, and they use an enzyme called reverse transcriptase to help with that. Viral medications are very difficult to develop for a reason that we'll talk about, and but essentially, almost always, with a few exceptions, we have to rely on our own immune system, help our own immune system defeat these things. Just a little bit about what people are calling coronavirus right now. As I write this, the official name for it right now is SARS-CoV-2. And it's part of a family of viruses. What the first S stands for is escaping me right now. Acute respiratory syndrome, I think, is the last three. These things have caused outbreak in the past. This is a new one, a novel one. That's another thing you hear people call it, like novel COVID-19 or whatever. Infection comes from droplets produced by an infected person's sneeze. It seems to be the case that this virus can remain viable on surfaces for several days, depending on the type of surface. It uses the glycoprotein spike. It attaches to an enzyme that's in the lungs. So this coronavirus specializes in the lungs, and it uses an enzyme called ACE2, an enzyme that's on the surface of these lung cells. And that's how it gets in. That's how it gets into the cell. This could potentially be a means of treatment. If you get a serious infection with this, what it does is, it causes breathing problems and all that, but it also basically depletes the resources of your immune system, and then other things, bacteria, who normally couldn't get a foothold, will come in, and often that's what will cause people to pass away, is a widespread bacterial infection that results from that. People are still trying to figure out how lethal is it, depending on the age group and stuff that you look at. Current information that I was looking at coming from China, death rates are about 0.2% to 15%, depending on how old you are. There's a Kurt Gazat, if you're familiar with that. It's a YouTube channel on the coronavirus, which I think is pretty interesting to look at if you wanted to learn more about it. So what's the deal with viral infections and why are they difficult? Well, for one thing, there's not much to them. Like I was saying before, with the bacteria, they have something very clear that differentiates them from our own cells, just that they have cell walls. This is not the case with a virus. They just have a little bit of protein, a little bit of genetic material, and all that. When they're actually actively reproducing, they're inside the host cell. So again, thinking of bioavailability and all that, how are you going to target something that's inside the cell? They multiply very quickly, so there can be trillions of virus particles all around the body before really you even know what's going on. They mutate quickly, so they can make certain treatments you come up with ineffective fairly quickly because they're so quick to mutate. So what can you do? Well, maybe you can alter the cell's DNA somehow so the virus can't use it to multiply, block enzymes that let it get in the cell, so basically try to prevent it from getting in or out of the cell, or stop it from leaving, or stop it from multiplying itself if you could target certain enzymes. So basically the general gist of a virus is you got a virus that sort of docks, as they say, with the host cell. It goes inside, it releases its genetic material. If it's a retrovirus, it's going to be RNA or it's DNA. And understand that everything that happens inside a cell, it's just a chemical reaction. The machinery of the cell is going to do what it does. Whether it's your own DNA, whether it's your own RNA, if it finds some instructions to assemble proteins or something, it's going to start doing it. And it's going to unknowingly, maybe, the machinery of your cell, start building viruses instead of what it's supposed to be building. And then when you get a bunch of them in there, they end up bursting out and go on and infect the next cell. So hemagglutinin is a virus, the protein that it uses to help dock to the cells. I don't know if that's the specific one that the COVID-19 virus does. Neuraminidase is an enzyme that helps it leave the cell. Sialic acid is the substrate of this enzyme, the thing that it works with, and that basically triggers the cell membrane to open and let the cell out. So these are both things, again, if we're looking for something that our cells don't really use, but viruses do, these are both areas that you could try to attack or utilize. It turns out that neuraminidase is a more effective route. So there's two anti-flu. Tamiflu and Relenza are the kind of the brand names of those, and they're taken, they're really only given to people who have the flu, and as to my knowledge, these don't work with coronavirus. Maybe they do. To my knowledge, they don't. But you have to take them in 48 hours. Even with the regular flu, you wouldn't give it to somebody unless you really thought they were susceptible of dying from the flu. And they act as competitive inhibitors for neuraminidase. This is the actual substrate. This is the Tamiflu. So, definitely certain similarities, similar enough that it can be a competitive inhibitor. IB singles out HIV and AIDS is something that you need to know about. It was first recognized in 1981 and caused by the HIV virus. So AIDS is the disease. HIV is the name of the virus that causes it. It causes a couple million deaths a year. I was actually looking. They thought maybe about 1 million people had died from it last year. So maybe those are slowing down compared to when I looked at this at first. So what the HIV is, or what it does, is it is a disease that specifically attacks the immune system itself. These cells called helper T cells is kind of what they're commonly called. CD4 T plus cells is kind of the more official name of it. So they, like all viruses, they dock to this, they penetrate the cell. It's a retrovirus. They use reverse transcriptase to make DNA from RNA. This is a virus that can remain dormant for many years sometimes, and then finally end up causing infection. But basically, the viral DNA just kind of integrates itself right into the actual DNA, and then the cell is just going to go ahead and make virus particles, and then when the cell dies, you release a bunch of HIV viruses. So what's the deal with HIV? Why is it so hard to combat? And when I was your age, having AIDS was definitely thought of as a complete death sentence. Like you were going to die from it eventually. It's not thought that way anymore, at least not in America. But the rest of the developed world, I should say. Basically, what happens is it's just, as I said before, we really need to rely on our own immune system to defeat viruses for the most part. So this is actually disabling your own immune system, and it's not really the HIV virus itself that kills you. It is the other things that come in, and that's how AIDS was kind of first discovered, as people were getting these weird illnesses and infections that just nobody ever should be getting. It's because they basically had no immune system. So it's attacking the very thing that can, combat it, so that makes it hard to combat. It mutates very rapidly, even compared to other viruses, even within a patient. It lies dormant within host cells, as I said, for years, so there's nothing for the immune system to respond to. So there is, basically, if you have the money, you can remain alive indefinitely, as far as we know, with HIV. They've developed a so-called cocktail of drugs, a group of drugs in combination. The problem is it's pretty expensive, about $12,000 annually. There hasn't been any success in developing a vaccine, as far as I know, due to the quick mutation. AIDS is a big problem in Africa for various societal reasons, being many of them. There's a high rate of infection there, and they may not have the funds, the availability to use this so-called drug cocktail and cure themselves, and so majority of those 1 million people that are dying from AIDS are from less developed countries. So that's it, folks. I know this was definitely not something to take your mind off the situation at hand. In fact, it was just the opposite. But sometimes knowing a little bit about what's going on can actually help reduce some of the anxiety. And yeah, this one got a little long, but thanks for watching, folks, and I hope to see you again soon. Bye-bye.